Welcome to another video. Today I'll be discussing about another disease named as the botulism, which is important for human, livestock and birds. Let's talk about the first of all etiology. The disease this is caused by the Clostridium botulinum. This botulism this is a poisoning caused by a toxin that is produced by this bacterium This is called the Clostridium botulinum. And this toxin may be most commonly taken up by orally, intoxication, or it may be produced inside the digestive tract during the infection. And botulism is a neuroparalytic illness resulting from the action of a potent toxin that is produced by this organism. So let's see Clostridium botulinum. Clostridium botulinum, that is a gram-positive bacteria. This gram-positive means the bacteria which are taking the ground staining. These are called the gram-positive bacteria. So it's one kind of gram-positive bacteria, and it is also anaerobic bacteria. We know that which are anaerobic bacteria. The bacteria which can be living with the presence of the oxygen. These are called the anaerobic bacteria. So it must need the anaerobic condition for its survivability. Then this bacteria, this is actually spore forming. Spore forming bacteria, these are actually very long living. They are very long living. Why they are actually very long living? Because in the adverse situation or adverse condition, they actually make the spore and they actually ultimately protect themselves, and they actually just increase their survivability against the adverse condition or the bad environment. Okay, and this is also the saprophytes. This actually, what does it mean by saprophytes? The saprophytes are those bacteria or those organisms which actually grow on the decaying or putrefactive materials or decaying materials. So these are this is also one kind of saprophytic bacteria. Then this is rod-shaped. According to its morphology, this is rod-shaped bacteria. Let's see that how Clostridium botulinum, why actually the name is Clostridium botulinum. Clostridium, actually this name came from the cluster, that means the spindle. That means the bacterium actually spindle appearance. That's why it is called the Clostridium, and it's a one kind of it's the name of the genus. So the genus is Clostridium. All the bacterium from this Clostridium genus, these are this name actually came from the spindle. That means actually these are the spindle-like bacteria. Okay, and the second name, that is a species, botulinum. So there is a history for that. The recorded history of botulism that began in 1735, when the disease was first associated with the consumption of the sauces. And botulism, the word actually came from the Latin word botulus. This Latin word botulus means sauces, and the name was kept as the botulism by a German doctor, and his name is John Müller. He is basically a German physician, and he just kept this name botulism in 1870. Then the disease was first described by Amenjem in 1897 after the investigation of a foodborne outbreak in Belgium. So finally we got the name, that why it is called the Clostridium botulinum. Clostridium means spindle, and this is the genus, and botulinum, this came from the word botulus, which means actually sauces. And the history we learned that actually from foodborne pathogen by eating the sausages, it was firstly found. That's why it is actually named as the sauces. Okay. Now let's talk about their toxins, their missiles. So let's think about that. They actually having the 4 missiles. Okay, the first missiles or first toxin. This is actually the preformed toxin. The first one, this is actually foodborne botulism. This is actually called the foodborne botulism, and that results when there is an ingestion of the food which actually containing the preformed toxin. That means actually just before the ingestion of the feed, actually this remains within the feed in the preformed. Okay, so this is actually the foodborne botulism is rare, but it may kill rapidly and contaminate the products, expose many persons, and it actually having the medical significance. Number 2, missile. If we just talk about this, this is actually the owned botulism, W for owned, and suppose this is toxin number 2. So the owned botulism is the second form of the botulism that actually caused by the organism that multiply and produce toxin in the contaminated owned. If the animal or human actually having owned, and if this is contaminated by the Clostridium species, Clostridium botulinum, then obviously they actually can have this toxin. So this is actually produce the toxin within the owns. That's why it is named as the W or own toxin. Then number 3, that is the infant botulism, which is designated as the I here. So infant botulism, this actually caused due to the production of the toxin by germinating the spore of the Clostridium botulinum in the intestine of infant. That means first of all it's actually going to the intestine of the infant. After going to the intestine of the infant, they actually cause the germination. They actually go in the form of the spore. They just move to the intestine and they germinate there. There from the spolation, they actually go for the vegetative form. After going to the vegetative form, they actually release these exotoxins. Okay, this is that's why this is actually called the like infant form of botulism. Then the fourth one, that is the child, or this is called the adult botulism, which is designated as the A here, and this from the intestinal colonization, that is represented by those cases in which the no food vehicle can be identified. So here actually we observed total 4 forms of botulism. Number 1, that is the preform form of botulism, which actually the foodborne botulism. Then number 2, this is the owned botulism, that actually spreading because of the owns in both humans and animals, I mean in the livestock. Then in this condition, actually this is called the infant botulism, which is actually occurring in infant. This actually first of all going to the intestine. After going to intestine, they actually causing the germination from this sporulation condition. They actually getting turned into the vegetative form, and they actually releasing the exotoxins, and these exotoxins are causing the destruction. We'll be talking at later about the mechanism. Then the last one, that is the this form is called the adult form, or this is the sometimes it is also called the child form. This actually they do the intestinal colonization first of all, then ultimately they actually cause the destruction by their exotoxin release. Here we'll be learning about the pathophysiology of this disease, and this is a very clear illustration. So if we think about this is the Clostridium botulinum, this toxin, this bacteria actually it's having the plasmid here, actually containing the gene, the toxin gene, which actually just producing the toxin, and this is one kind of exotoxin. We know that gram-positive bacteria, they actually release the exotoxin, whereas the gram-negative bacteria, they actually release the endotoxin, and endotoxin are basically released after the death of the organism. Now as this actually this gene, this toxin gene actually synthesizing the toxin, these toxins are getting expelled out by the exocytosis. These are getting expelled out. After expelling out, as I mentioned that these Clostridium species, these are commonly found in the soil, and these are also saprophytic bacteria, and they remain in the sporic form. So whenever they actually having the anaerobic condition, or the ambient temperature like 15 degree to 35 degree centigrade, they can produce the toxin. As they can produce the toxin and they are releasing the toxin, if the toxin actually contaminate the animal feed, we know that animal actually go for the foraging. They also sometimes the feeds and forages which are kept stored for a long period of time, and after that they are given to the animal for their ingestion, for actually having. But in this condition, if the toxin actually doing the contamination of that, so if it's doing the contamination, then what will be happening? That it will just go it will to the animals directly to the intestine by the oral route. It can be one source, one way. Okay, another the carcass, any carcass, actually the decaying carcass, by this carcass or like the fly maggots. These are also very common for spreading of this bacteria. So this can be also another source. So carrion can be also another source, or like carrion of bone, or the animal actually which having the pica or leaking syndrome, they actually leak anything what actually not ingestible. Okay, they actually leak due to the mineral deficiency like phosphorus deficiency or copper deficiency. For those actually there will be the leaking syndrome. So if in this leaking syndrome the animals actually leak anything, so if they actually leak and they are actually remaining the toxin, so toxin will be just orally will be introduced in the animal. As well as if the animal actually having any wounds, as I mentioned that the Clostridium, this Clostridium can be occurred in 4 forms. Okay, one of them is wound form, wound botulism this is called. So if the animal actually having any wounds, from that wound that bacteria can easily pass through. Okay, now let's think about this, that by the animal feed or like carcass or like fly maggots or due to leaking syndrome or anything, for any causes actually this is orally introduced. Whenever the toxin or the organism, anything can be as I mentioned that it can be the toxin for preformed toxin form. The first one, foodborne botulism. In foodborne botulism we described. So if you think about this, that a toxin is the animal actually ingesting the toxin, or like the bacteria can be also directly introduced inside of the oral cavity, inside of the stomach. Now if it is introduced orally, this is going to the stomach, and simply I just drawn this figure. So after just going, it will just move to the intestine. Okay, after moving to the intestine, what will be happening in the intestine? Either the toxin if this is in the preformed toxin, so what will be happening if we just make this figure a little bit bigger, so we'll understand that from this intestine actually, if the preformed toxin we actually have the animal actually have, then the preformed toxin will be directly absorbed into the blood vessels. Okay, and if the actually the bacteria just going to the intestine, if the bacteria actually present in the intestine, so the bacteria first of all it will be just moving for the germination process. I mean from the spore form, this will be now converted into, it will be converting to vegetative form. Now this vegetative form, this bacteria right now will do the release of the different types of exotoxins. Okay, like commonly C and D, which are responsible for doing the toxicity. So this bacteria is now supposed releasing the toxins. So if this is releasing the toxins, those toxins will also be absorbed by the blood vessels. Now what happened exactly here? This is called the toxemia. Why it is called the toxemia? Because toxins, these are present in the blood. That's why it is called the toxemia. Now from this just via this blood circulatory system, this will be circulating in the animal body. They will reach to the postganglionic nerve fibers. Now let's actually understand a little bit deep. If this is the lateral view, if you think this is the lateral view of the brain spinal cord. Okay, and we know actually this is the what? These are the parasympathetic fibers. We know the parasympathetic fibers actually, these are given the parts. These actually parts in the muscles. Okay, and this actually doing the what? Contraction and relaxation of the muscle. These are the basically craniosacral outflow. So these parasympathetic fiber, these are the preganglionic parasympathetic fibers. These fibers, these actually, we know that parasympathetic fibers, these are the craniosacral outflow. Though actually in this picture, it was actually shown, this actually I shown from these fibers actually originating from here. But actually the fibers actually are originating from the cranial region as well as from the sacral region. Now what I'm mentioning that from these actually fibers, these are called the what? The preganglionic fiber. These are just going to the and terminating to the ganglion. Okay, after ganglion there are the what? there are just flowing the postganglionic fibers, and whenever the postganglionic fibers are getting flowed, they are actually fibers in the muscle. They actually are responsible for the muscular contraction and relaxation, as I mentioned before. So we'll be just making it more bigger to understand what's happening actually, what these toxins actually doing here, why actually in botulism we know that there will be the flaccid paralysis. Now why actually this occur, we'll be just learning it a little bit deep. So if you think this figure, this is actually the figure of a neuromuscular junction. On the right, in this figure, this is the neuromuscular junction. Normally in the neuromuscular junction, what happens? We know that in the presynaptic membrane, there are some preformed vesicles, and these vesicles containing acetylcholine. Thus acetylcholines are remaining within the vesicles. Now whenever actually there are any action potential, there is actually generating any action potential, these actually come and they actually call the cell depolarization. From the resting membrane condition, they actually doing the cell depolarization, and due to the cell depolarization, what is happening here, that this acetylcholine, this containing actually these vesicles containing the different as the snare proteins. Okay, these snare proteins are responsible for the fusion of these vesicles with this presynaptic membrane. Now whenever they actually doing the fusion, what is happening, that this vesicular membrane, this actually doing the fusion with this presynaptic membrane due to the snare proteins, and that's why the acetylcholine, this is released outside. Whenever this acetylcholine is released outside, and this will be accepted or received by the what, the muscle fiber, I mean the sarcolemma of the muscle. I mean we know that this is called the postsynaptic membrane. Okay, this postsynaptic membrane, so they will be received by that. Whenever they will be received by that, there will be what, the muscle will be contracted, and muscular activity will be continued. But we know that actually this toxin only affect the cholinergic neuron. We know that these are the what, the cholinergic fibers, these are the cholinergic neuron, these are not adrenergic neuron. It should be it is actually very much important to keep remember that this only work on the cholinergic neurons, this will not act on the adrenergic neurons. So it is very much important. Whenever these actually are getting saturated, the receptor in the muscle fiber which are getting saturated with the cholinergic with the acetylcholine, so they actually doing the what, the impulse transmission. Now if you think about this, like the toxins, what these toxins actually doing, they are circulating. After this circulating, they actually come to the just surround to this neuromuscular junction. Now in this neuromuscular junction, what they actually do, they have the capability for easily passing through. It is important. They actually having the heavy chain, light chain. So by this heavy chain, actually they are doing the fusion with this membrane, with this fusion with this membrane. Now whenever actually they are doing the fusion with this membrane, they actually are endocytized. Whenever they actually are endocytized, what they are actually doing, they actually attacking the V snare proteins. As I mentioned, that snare proteins are responsible for the fusion of the acetylcholine with the presynaptic membrane. So these snare proteins are attacked by this toxin. Now whenever these are attacked by the toxin, now what will be happening, the snare protein will be destroyed, snare protein will be deactivated, remaining deactivated. So the vesicle, what will be happening, the acetylcholine filled vesicle, they will not do the fusion with this presynaptic membrane. As they are not doing the fusion, what will be happening, the acetylcholine will not release. So if the postsynaptic membrane, if the muscle actually not getting the acetylcholine, so what will be happening? If this is not getting acetylcholine, there will be no impulse conduction, there will be no nerve impulse transmission, so there will be no contraction, no response of the muscle. So that is the thing, that is the how actually this toxin actually causing the paralysis, because as the nerve impulse actually not getting conducted, so what will be happening, the nerves will be the muscle will be paralyzed. So we got the main thing that actually this actually is the parasympathetic blockings. Okay, that's why there will be the paralysis. Now there are like spastic paralysis and flaccid paralysis, different types of paralysis. But what does it mean by flaccid paralysis? Now flaccid paralysis means exactly that whenever actually your muscle get weaker and weaker due to the paralysis, this is actually called the flaccid paralysis. When the muscular weakness are observed, this is called the flaccid paralysis. That means your muscle tone actually, these are actually lost and these are actually toned off. Okay, these are lost, and that's why this is called the in the muscular weakness, this muscular weakness is called the flaccid condition. Okay, and the paralysis occurring, that's why it's called the flaccid paralysis. Okay, then this flaccid paralysis, this become more severe condition. Like first in the primary condition, in the beginning of this, we know actually this toxin, the botulism toxin, this is actually within a very short dose, actually it can do the more destruction, more actually it can do the effect on the body. What will be happening, that muscle will be, different muscles will be start to get paralyzed. Okay, it may be like eye muscles, it may be like pharyngeal muscles or laryngeal muscles. Then it also affect at one time, actually it will affect the respiratory muscles or cardiac muscles. Now if think about it, that respiratory muscles are getting seizing, these are getting cyst, these are not working, there is the paralysis. So what will be happening, there will be respiratory failure. If you think about the cardiac muscle is not pumping due to the paralysis, flaccid paralysis. So what will be happening, the cardiac muscle will not work. So if the cardiac muscle will not work, your total circulatory system will be stopped. So these are the things, which are responsible for the death of the animal. Okay, so if the load of the toxin that very heavy, as I mentioned before, that this toxin within a low dose, actually with a low dose, it can actually cause the more effect on the body. So if the load of the toxin is very heavy, the animal obviously will be dead within the 24 hours. Okay, but if the toxin load is less, then obviously it may show the signs and symptoms, or like it can do the it can show the symptoms actually within 3 to 7 days. In the animals, within 3 to 7 days the symptoms will be observed. Now usually what happens, that first of all, how you will understand that actually the animal will not try to eat because it's having the paralysis, and it will not try to eat, even though like drinking become impossible for this animal. So this is actually one symptoms by which we can understand it is actually not a contagious disease, it's not transmissible. How it's occurring, I already mentioned it. Let's talk about the diagnosis. What can be the way of diagnosis? We can do the diagnosis on basis of symptoms. As I mentioned that in earlier stage, if the load of the toxins is heavy, then obviously the animal will be dead within 24 hours. Less than 24 hours they will die. But if the toxin is less present in the blood, so there will be what within 3 to 7 days the animal will start to show the symptoms. And first, as I mentioned, the symptoms will be like due to the flaccid paralysis, everything will be like every muscles. Okay, the muscles will be start to get paralyzed. So the affected animals, they will maybe weaker. Also, they will be stagger about or go down. They will show the recumbency, as well as the cattle. This cattle actually characteristically they actually display the flaccid paralysis, and occasionally the protrusion of the tongue. It is one of the most important. That protrusion of the tongue will be observed easily. In most cases, the disease is fatal, although some animals may recover. In many cases of botulinum, then we can do the ELISA, enzymatic immunosorbent assay. By this ELISA, actually we can identify, or we can actually check the antibody condition. If the toxin is actually present, there will be the antibody production, and the antibody will be found in the serum. So we can actually test the serum to get the result, actually if the toxin is present or not. Then we can do the bacterial culture. If we do the bacterial culture, then we'll just get it that if this is positive or not. We can do also the mice testing. If we just take the samples from the animal, if just we do the inoculation in the mice, so if the mice is getting died, so if the mice are getting died, then obviously we have to understand the presence of the toxin. We can also do the we can also diagnose the disease by necropsy finding, such as in the most of the cases actually there will be the hemorrhage. Actually there will be the hemorrhage. Severe enteritis can be occurred because, as I mentioned, that the germination of the if the toxin actually present in the intestine, or like the bacteria actually that is actually present there, so they were actually producing some sorts of toxins there after the germination. So after the germination they are producing the toxin, and these toxins actually are causing the enteritis. I mean hemorrhagic, there will be the hemorrhagic enteritis. Okay, and also the heart muscle can be affected. They can also have the pulmonary edema, then congestion can be noticed in different parts of the body, as well as the excessive pericardial fluid containing the free-flowing fibrin. This may also seen, but most characteristically we can see in the hemorrhage will be in the intestine. So this is actually we can see. Then we can also do the antibody detection by different ways. Also we can do the isolation of the bacteria, and by the isolation we can easily understand if the toxins, if the bacteria actually present or not. In the bird, this flaccid paralysis actually, this also occur in the bird, and the bird actually they actually droop their head down because of the neck muscle paralysis. Because of the laryngeal muscle paralysis, neck muscle paralysis, they actually go for their head actually got drooped down, and this actually called as the limber neck. So how we can do the prevention and control? We already just understood the course of the disease. So we know that if we just give the animal fresh preserved food, feeds and fodder, then obviously it can be easily controllable. As well as we can also do the vaccination. Vaccination is available in some countries, so we may also do the vaccination. By this vaccination we can prevent the disease. The farmer should not provide any preserved forages for the animals which have been rotten or which actually preserved for a very long period of time. Then also if the animal actually having any wounds, then obviously we must treat the wounds carefully so that the toxin can't do the wound infection. If we can do this, then obviously we can prevent and control the disease easily. So I hope you understand all of this, that how actually the disease is occurring and why it is called the Clostridium botulinum, what are the toxins of them, how actually the toxins affecting the nervous system, and why the paralysis is occurring, and what will be the diagnosis, prevention and controlling strategies. I hope actually all of you understand. Thank you.